Good morning, dear students. So in today's class, we are going to discuss an interesting topic, which is genetic engineering. Before going in detail, let's see some basics. Okay, students, you all possess several characteristics exactly as your parents. Maybe your nose, eyes, mannerisms, gestures, anything else. So what may be the reason for the similarities? So the reason behind the similarities is called heredity. And there are differences also. You are not exactly as your parents, and which makes you unique. So the differences are called variations. So the differences is called variation and the similarity is called heredity. So the branch of science that deals with heredity and variation is called genetics. The fundamental unit associated with genetics is genes. So genes are the structural and functional unit of life. These genes can be modified to get several desired characteristics. So this modification is called genetic engineering. We have several applications which may be used in crop improvement program, plant breeding, etc. So let's see some of the application. And the first one is the production of human insulin using recombinant DNA technology. So recombinant DNA technology is a technology that we use in genetic engineering. So before going in detail, I will give you a general outline about the recombinant DNA technology. You consider there is a gene of interest. By gene of interest, we means a gene which produces a desired protein. So we have a gene of interest in a particular chromosome in a cell. So we have to cut it from there. For that, we use a specific enzyme called restriction endonuclease to cut the gene of interest from that chromosome. And now we have to integrate this gene of interest to a vector. A vector is generally a vehicle, a DNA vehicle. A vehicle used in genetic engineering to carry something to somewhere. So we have to cut the vector also, use the same restriction endonuclease to cut the vector, and now we insert the gene of interest to the vector and integrate it with the help of another enzyme called DNA ligase. Now we get a recombinant DNA. DNA can be inserted in the bacteria, allow it for multiplication, and after a series of processes like transcription, translation, we get the desired proteins in bacteria or the host cell. Later, this protein can be extracted and purified and can be used. General outline of recombinant DNA technology, and let's see how this recombinant DNA technology works in the production of human insulin. So, genetic engineering. So as I said, genetics is the branch of science that deals with heredity and variations. So this is a proper definition for genetic engineering. Genetic engineering is the technology of controlling traits of organisms by bringing about desirable changes in the genetic constitution of organisms. Moving on to the production of human insulin. Here you can see this is the human cell which contains the human DNA. This is the enlarged version. So this red color portion is actually the insulin gene that contains in the human DNA. At the same time, there is a bacteria which contains a bacterial plasmid. So this plasmid is enlarged here. The plasmid is actually an extra chromosomal DNA molecules, a molecule which autonomously replicate, which have the capacity to replicate independently in a host cell. Now we have to cut the insulin gene from this chromosome using restriction endonuclease, and we have to insert it into this vector. So as I said earlier, the vector also want to cut with the same restriction endonuclease, and we integrate the insulin gene with this plasmid, and this version is called the recombinant DNA. Now this recombinant DNA is inserted into the bacterial cell. So here this is the bacterial cell, and we insert into this recombinant DNA into the bacterial cell, and the bacteria is allowed for the multiplication. So along with the transcription and translation, the proteins, the insulin gene is expressed and insulin is produced within the bacterial cells. So this is the bacterial culture which contains the insulin, which is then extracted and purified and finally bottled like this, which can be used. So this is the production of human insulin using genetic engineering. Now let's see another application of genetic engineering, which is BT cotton. So I hope you are familiar with cotton plant, which has high economic value. The cotton can be used in many industries. But there is an insect which affects the growth of cotton plant and which ultimately result in the reduced yield of cotton. So there should be a solution, right? So a solution for this was erased via genetic engineering. Okay, so you might have thought why this picture, why this picture of a bacteria, Bacillus thuringiensis, is shown here, right? So this is a bacteria called Bacillus thuringiensis, which has a protein called cry protein. This cry protein is actually a toxin which produced by cry gene. But this toxin or the cry protein is not toxic in the bacterial cell. Okay, I repeat, this toxin or the gene or the cry protein. The cry protein is not toxic in the bacteria, but it is toxic in an insect. There is no need to remember the name of the insect, but you just remember it's an insect which affects the cotton plant. So the Bt toxin or the cry protein is activate in the insect gut in an alkaline pH. So how we can use it? This is the question. So by using the recombinant DNA technology, we isolate this gene from bacteria and insert it into the DNA of the plant cell. Now, when the insect come and feed on the plant, the cry protein get into the insect gut and activated there. As it is a toxin, the insect will die. Thus, we made a solution for this infection using genetic engineering. So there are a lot of applications like, which can be used in the crop improvement program, like the production of flavor saver tomato, which aims to increase the shelf life of tomato, and golden rice, which aimed to reduce the vitamin A deficiency, etc., which can be discussing later. Moving on to another concept, which is gene therapy. So, why does we go for gene therapy? Students, we know that the change in the structure and function of genes are responsible for various diseases, right? So, if those genes are identified and removed, we can get rid of that disease. Am I right? But the question is that how can these genes that are responsible for disease be identified from the thousands of microscopic genes? There are thousands and thousands of genes. So how we identify the genes that causing the particular disease? So here comes the importance of Human Genome Project. Are you familiar with this term? Human Genome Project. So by the name genome, we actually mean the entire set of genetic material present in an organism. So in 1990, Human Genome Project was started, and by a technology called gene mapping. By a technology called gene mapping, here you can see gene mapping, we identified the location of genes that are responsible for various traits. So as per the current syllabus, the molecular mechanism or technology behind the gene mapping is not there for your study. You just remember the terminology gene mapping. Okay. So by using gene mapping, we can identify the gene causing undesired characteristics and would able to remove that. So now let's look what are the major findings of Human Genome Project. So the human genome has about 24,000 functional genes, and major share of human DNA include junk genes. By junk means non-functional. So there is only 0.2% difference in DNA among humans, and about 200 genes, that is really surprising, about 200 genes in human genome and identical to those in bacteria. So these are the major findings of Human Genome Project. So what are things that we learned today? The production of human insulin using genetic engineering, especially or specifically saying the recombinant DNA technology, gene therapy and Human Genome Project. So this is all for today. Hope you understand the topic. See you with another video in the next class. Thank you.